You know those moments when you just feel okay? Like there's enough air, you're safe, it seems simple.
Mhm.
But what's actually happening inside your body, down at the molecular level, to give you that feeling? It's just
Yeah.
incredibly complex.
It really is. And that's kind of what we're digging into today, but in a much simpler organism.
Right. Okay, so let's unpack this, because today we're doing a deep dive into something pretty fundamental. How this tiny little worm, Caenorhabditis elegans, actually senses the oxygen around it.
And it's fascinating because, even in this worm, the whole setup is remarkably sophisticated. We're going to focus on one particular signaling molecule, cyclic GMP.
cGMP.
Yeah, cGMP, and specifically its role in one key nerve cell that senses oxygen called PQR.
Exactly. And the way we know this stuff is pretty amazing. It comes from a scientific study where they used these incredible new techniques.
Yeah, cutting edge stuff.
Seriously,
Yeah.
imagine being able to actually watch molecules doing their thing inside a living worm
Mm-hmm.
and even tweak them. That's basically what they did with cGMP here. So our mission for this deep dive is to figure out the key bits and pieces, and maybe even more interesting, these feedback loops involved in sensing oxygen.
And there's a twist coming.
Oh yeah, get ready for this. Even worms that are genetically identical, well, they don't always react the same way.
Let's jump right in with the star player, cGMP.
Okay.
So for anyone maybe not familiar, cGMP is what's called a second messenger.
Like an internal memo inside the cell?
Pretty much, yeah. It relays signals, and it's involved in a ton of things in nervous systems, sensory stuff like vision or smell, how excitable neurons get, even learning and mood in, us.
Right. But actually seeing it change in real time in a living animal, that was tough for a long
A
time.
major challenge, yeah. You can't just plug something in.
But that's where these new tools come in. They developed this genetically encoded sensor, CGA 500.
That's the one, cGA 500. They engineered the worms so this sensor would be produced right inside the neurons they wanted to study.
And it glows differently depending on how much cGMP is there.
Exactly. More cGMP, different glow intensity, so you can literally watch the levels change.
Wow. Okay.
And they put this sensor specifically into the worm's main oxygen-sensing neurons, AQR, PQR, and URX. But like we said, we're really focusing on PQR today.
Got it. So the experiment itself, what do they do?
It was actually quite elegant. They took these worms, put them in a controlled environment, and then changed the oxygen levels.
Okay.
Specifically, they'd shift it from, say, 7% oxygen up to 21%, which is like normal air, and then back down again.
And watch the sensor in the PQR neuron the whole time.
Precisely. Watched how the cGMP levels flickered up or down in response.
Okay, so the results. This is where it gets weird, right?
Yeah, this was the first big surprise. When they increased the oxygen, the cGMP level in PQR didn't just do one
It
thing.
wasn't consistent.
Not across different worms, no. In some worms, cGMP went up, which, kind of makes intuitive sense. More oxygen, maybe a stronger signal.
Sure.
But in a whole bunch of other worms, remember, genetically identical, raised the same way the cGMP levels actually dropped when oxygen went up.
Whoa. Okay, that is a curveball. How does that even work?
Right. It immediately throws up this question. How can identical individuals have such different internal responses to the exact same thing?
But wait, there was consistency within a worm, wasn't there?
Yes. That's the other fascinating part. While the direction was different between worms, some up, some down, any single worm was incredibly reliable.
So if a worm was an upper, it always went up.
Every single time they repeated the oxygen increase on that specific worm. Same for the downers. Each worm had its own, signature response.
That's amazing. Did they quantify that?
They did, and the numbers are pretty stark. Get this. Almost 97%, 96.9% to be exact, of all the variation they saw in the cGMP response was down to differences between the individual worms.
Only about 3% was variation within the same worm over time.
Exactly. Tiny. So huge differences between individuals, but really consistent responses within each one.
And they saw something similar with calcium signaling, too.
Yeah. They looked at calcium changes in the same neurons, and again, high variability between worms. About 88.5% of the variance was inter-individual, consistent within, variable between.
So identical twins, even in the worm world, might react quite differently on the inside.
It strongly suggests that, yeah, even with the same genes, same environment, there's room for significant individual variation in how neurons are tuned.
And the idea is maybe tiny random differences in how much of certain proteins get made.
That's the hypothesis. Stochastic fluctuations in gene expression could lead to slightly different levels of key signaling molecules, setting each worm on a slightly different path.
Okay, variability established. Let's get back to the basics, what actually senses the oxygen to start this whole cGMP thing.
Right. The study identified an enzyme called soluble guanylate cyclase, GCY35.
BCY35.
Yep. And it seems absolutely essential. They looked at worms engineered to lack GCY35.
Yeah.
No response, zero change in cGMP when oxygen levels changed in the PQR neuron.
So GCY35 is like the trigger, the primary oxygen sensor that kicks off the cGMP signal.
Looks that way, yes. It's the ignition switch for this pathway in PQR.
Did signals from other neurons matter much? Like, was PQR just listening to its neighbors?
Good question. They checked that too. They used worms that had problems sending signals between neurons, synaptic transmission defects.
And did that affect the cGMP oxygen response?
Not really, no. It suggested that these external inputs weren't the main driver here. It seems PQR is largely sensing the oxygen and changing its own cGMP levels directly.
Cell autonomous, they call it, doing its own thing.
Exactly. Relying mostly on its own internal machinery for this particular response.
Okay, so let's circle back to the up versus down puzzle. If GCY35 makes cGMP when oxygen goes up, why would cGMP ever go down?
Feedback loops. The researchers figured there had to be negative feedback involved.
Like thermostats. Something kicks in to counteract the initial change.
Precisely. The initial rise in cGMP, triggered by oxygen via GCY35, likely sets off other processes that then push cGMP levels back down, or at least temper the rise. Keeps things in balance.
So what's involved in that feedback?
A major suspect was calcium. Calcium ions, K2+4 plusr.
Okay, how does calcium get involved?
Well, the model they proposed goes something like this. Oxygen up, GCY35 active, cGMP goes up.
Right.
Then that increased cGMP opens specific channels in the neuron's membrane. These are called cGMP-gated channels.
Channels gated by cGMP.
Makes sense. And when they open, calcium ions, among other things, can flow into the neuron from outside.
So the initial cGMP rise lets calcium in.
Exactly. And that influx of calcium, they hypothesized, could then trigger downstream stuff that reduces cGMP levels. Calcium acts as the trigger for the break.
Do they have evidence for that? Like, what if you block those channels?
They did exactly that, genetically. They looked at mutant worms missing key parts of those cGMP-gated channels, proteins called TX2, TX4, CNG1.
And in those mutants.
The cGMP never decreased when oxygen went up, ever.
Wow. Okay.
And not only that, the cGMP increases they did see were actually much bigger, much stronger than in the normal worms.
So removing the channel, removed the brake, and also made the accelerator push harder in a way.
That's a good way to put it. It strongly supports the idea that calcium coming through those channels is essential for that negative feedback, for putting the brakes on the cGMP signal.
This sounds like it could get complicated inside the cell. Is cGMP just, like, sloshing around everywhere equally?
Probably not. And that led them to look at compartmentalisation. Can different things be happening in different parts of the neuron at the same time?
What did they find?
They managed to measure both cGMP and calcium simultaneously in PQR.
Tricky.
Very. And they saw that in those cases where the overall cGMP level in the main cell body went down with increased oxygen, there was a sharp rise in calcium in that same area happening at the same time.
Okay, so decrease in cGMP, increase in calcium happening together.
Which fits the feedback model. But it gets even more interesting.
How so?
This led them to think about nanodomains. Tiny, localized areas.
Nanodomains, like little zones within the cell.
Exactly. The idea is maybe cGMP is actually increasing right near the cell membrane where GCY35 and those cGMP-gated channels are located.
Near the action.
But at the same time, further away in the main part of the cell body, the overall cGMP level might be dropping because that calcium-triggered feedback mechanism is kicking in more broadly.
So you could have cGMP going up in one tiny spot, but down overall.
That's the idea. Highly localized signaling, like micromanagement inside the neuron.
Mind blown. Okay, so who are the actual molecular players doing this feedback? You mentioned calcium, but what does calcium do?
Right. Calcium needs helpers. Enter the phosphodiesterase, PDEs.
PDEs, I've heard of those. They break things down.
Exactly. They are enzymes that specifically break down cyclic nucleotides like cGMP. So they are crucial for turning the signal off or dialing it down.
And is there a PDE involved here that calcium activates?
Yes. The study focused heavily on one called PDE-1. It's the worm version of a mammalian PDE that is known to be switched on by calcium.
So calcium comes in through the channels, activates PDE-1.
And PDE-1 starts chewing up cGMP. That's the core of the negative feedback loop they identified.
Makes sense. Did the evidence support that? What about worms without PDE-1?
Just like the channel mutants, worms with mutations in the PDE-1 gene also lost that ability for cGMP to decrease when oxygen went up.
Okay. Strong evidence.
Very strong. It fits the picture. Calcium influx activates PDE-1. PDE-1 reduces cGMP.
Did losing PDE-1 affect the worm's baseline cGMP before the oxygen change?
Only slightly in some cases.
Yeah.
Which tells us there must be other PDEs around as well, managing the day-to-day cGMP levels. PDE-1 seems specialized for this calcium-triggered feedback.
Right. And losing it messed up their behavior too.
Yeah, their movement response to oxygen changes was altered, likely because the calcium signaling, which controls muscle activity indirectly, was out of whack without PDE-1's influence on cGMP.
And they double-checked PDE-1's function.
They did. They even tested the worm PDE-1 in human cells in a dish, confirmed it gets boosted by calcium. And interestingly, it could break down not just cGMP, but also another related molecule, cMP.
Tool specificity.
And crucially, if they put a working copy of the PD-1 gene back into the oxygen-sensing neurons of the PD-1 mutant worms.
Did it fix the problem?
It did. The negative cGMP responses came back.
Okay, PDE-1 is definitely a key break triggered by calcium. But you said there might be other mechanisms too.
Yeah, the data hinted that PDE-1 isn't the only way calcium causes cGMP breakdown. There's likely more complexity, maybe other PDEs or different pathways involved. It's rarely just one molecule.
Always more layers.
Yeah.
Okay, so that's PDE-1, the calcium-activated break. But you mentioned another PDE was important.
Yes, PDE-2. This is where things get really counterintuitive.
Okay, what about PDE-2?
So PDE-2 is the worm version of a mammalian PDE that can be stimulated by, wait for it, cGMP itself.
Okay, so cGMP activates an enzyme that breaks down cGMP. That sounds like another feedback break.
You'd think so. But the results with PD-2 mutant worms were baffling at first.
What happened?
In many of the PD-2 mutants, when oxygen increased, cGMP levels either decreased or didn't change much at all.
Wait. Losing an enzyme that breaks down cGMP led to less cGMP or no increase. That seems backwards?
Completely backwards. It suggests that, in this specific situation, PDE-2 is somehow normally helping to promote or sustain the cGMP levels.
How on earth would that work? An enzyme that destroys cGMP helps keep its levels up.
Okay, stick with me. The researchers came up with a clever model. They proposed that PDE-2 might be acting, maybe indirectly, to limit how active those cGMP-gated channels are.
The ones that let calcium in.
Exactly. So if PDE-2 normally keeps those channels slightly in check, it would limit the amount of calcium coming in.
Okay.
And if less calcium comes in, then the calcium-dependent breaks like PDE-1 wouldn't get slammed on so hard.
So PDE-2 regulates the regulator. By limiting calcium entry, it prevents PDE-1 from going overboard and wiping out the cGMP signal too quickly.
That's the hypothesis. It's acting like a meta-regulator, preventing excessive negative feedback.
Wow. Okay. Any proof for that?
Some neat genetic evidence. They looked at worms that were mutant for both PDE-2 and one of the channel's subunits, CNG1.
So no PDE-2 and the calcium channel's broken anyway.
Right. And in those double mutants, the weird cGMP decrease seen in the PDE-2 single mutants was gone. Instead, they saw strong cGMP increases when oxygen went up.
Which supports the idea that PDE-2's weird effect is somehow linked to calcium entry through those channels.
Exactly. It suggests PDE-2's normal job here involves modulating those channels or their downstream effects.
Okay, my head is spinning slightly. Let's add another player. PKG.
Right, PKG. cGMP-dependent protein kinase. Basically, another thing that cGMP can activate, besides the channels in PDE-2. The gene in worms is called AGL4.
And what happened in AGL4 mutants?
They looked remarkably similar to the PDE-2 mutants.
Really? Same weird cGMP decreases?
Often, yes, or no change, and also reduced calcium responses, just like the PDE-2 mutants.
Okay, that similarity must mean something. Are they in the same pathway?
Seems very likely. They found that putting a working copy of AGL4 back into the neurons fixed the cGMP response in the AGL4 mutants. And similar to the PDE-2 story, if they made double mutants, AGL4 mutant plus a channel mutant, or AGL4 mutant plus a PDE-1 mutant, those weird cGMP decreases were reversed.
So PKG AGL4 is also somehow involved in preventing that excessive negative feedback.
That led them to propose a pathway. cGMP activates PKG, PKG then perhaps stimulates PDE-2.
A little cascade.
Yeah. So cGMP, PKG, PDE-2 regulates channels, limits calcium at less PDE-1 activity.
Okay, I think I follow. So PKG and PDE-2 work together to fine-tune the calcium influx and stop the PDE-1 break from locking up too hard.
That's the model. And the fact that PDE-2 mutants and Eagle 4 mutants look so similar, and making double mutants of the two doesn't make things worse, supports the idea they're acting together in the same pathway.
Okay, deep breath. Let's try and summarize the whole proposed picture then.
Let's try. So oxygen comes along, activates GCY3536.
Step 1. Make cGMP.
All right. Then that cGMP does a few things. It opens the ion channels, letting sodium and, crucially, calcium into the neuron.
Step 2. Channels open, calcium comes in.
But cGMP also activates PKG and PDE-2.
Step 3. Activate the meta-regulator.
Yeah. And PDE-2, maybe boosted by PKG, seems to put a slight damper on the calcium influx through those channels.
Step 4. PDE-2 fine-tunes calcium entry.
Which in turn controls how strongly the calcium-dependent negative feedback involving PDE-1 and potentially others gets activated.
Step 5. Control the brakes, PDE-1. So you get a response, but it doesn't immediately shut itself down too hard.
Exactly. It's this intricate dance between the initial signal, the ion influx, and these multiple interacting feedback loops involving PDE-1 applying the brakes and PDE-2 regulating the brakes.
Mutually regulating negative feedback loops. Sounds complex, but maybe efficient.
Could be. And the researchers point out that in mammals, the versions of PDE-1 and PDE-2 are often found in the same neurons, too, suggesting this kind of interplay might be a common design principle in nervous systems.
But it's still a model, right?
Yeah.
Probably not the whole story.
Oh, for sure. They acknowledge it's likely a simplification. Biology is always more complicated. There are bound to be other molecules, other connections we don't know about yet. Science marches on.
Okay, so wrapping this up. What are the big takeaways from this deep dive into a worm's sense of smell? Well, a sense of oxygen.
I think, first, just the surprising complexity hiding in a seemingly simple process in a simple animal. It's not just on-off.
Definitely. And the elegant balance between these different enzymes, PDE-1 and PDE-2, playing opposing roles in feedback.
Right. One slamming the brakes when calcium rises, the other maybe easing off the brake pedal a bit to allow a sustained signal.
And the whole idea of compartmentalisation, nanodomains, that signaling isn't uniform inside the cell.
Different things happening in different places. And then, of course, the big one, the variability.
Yeah, the genetically identical worms can have fundamentally different baseline responses, some increasing cGMP, some decreasing. That's still pretty wild.
It really highlights how much intricate tuning goes into even basic sensory systems and how feedback is absolutely critical.
And it also just shows that individuals, even clones, aren't necessarily identical in function.
Exactly, which leads to that final thought to chew on.
Right. We should leave people thinking, why? Why have this variability built in? If you've got genetically identical worms, why wouldn't they all react the same, presumably optimal way?
Could it be adaptive? Maybe it's like hedging your bets. If the environment changes slightly, maybe the down responders do better sometimes and the up responders do better other times.
So the population as a whole is more robust. Some individuals are tuned for slightly different conditions.
It's a fascinating possibility, isn't it? What looks like random noise between individuals might actually be a feature, not a bug, at the population level.
Definitely something to mull over. It shows how studying these tiny worms can give us really profound insights.
Absolutely. These fundamental principles of how neurons signal, how they use feedback, how they manage internal messengers like cGMP, these are likely conserved across the animal kingdom all the way up to us. It's a peek under the hood of how nervous systems work.